electrons to a bank where you can exchange them for ATPs. And I've got a long definition for oxidative phosphorylation. I'll give you the details just in a few minutes. So let's go back to what happens with these reduced coenzymes that we've generated in the Krebs cycle. That's where the real money is. That's where we're going to get most of our ATP. And it's kind of interesting to think about it. Each one of us receives our mitochondria from our mothers, not from our fathers. So the egg has mitochondria, the sperm does not donate any. And so our mitochondria, our powerhouse, so to speak, is a maternally derived structure in our bodies. A pretty important one, too, if you think about it. Now, let's see, what... My E, good. So we have generated lots of reduced NAD and reduced FAD, the NADH and the FADH2 from the Krebs cycle. Are going to donate their electrons to the electron transport chain. They donate these high energy electrons to the ETC, that electron transport chain. The electron transport chain is located in a particular spot. I'm drawing the mitochondrion now. And where is the electron transport chain? It's embedded in the inner mitochondrial membrane. So here's the ETC. Not the outer, smooth one, but the inner one that's thrown into these deep folds. That's where the electron transport chain is, the electron transport system. Right? And the Krebs cycle enzymes, they're located in the matrix here, this fluid portion in the central area of the mitochondria. So they're in close proximity to each other. Okay. And what happens in the electron transport chain is that the electrons delivered by NAD and FAD are passed. These electrons are passed from one electron carrier to another down this electron transport chain. From one carrier to another down the electron transport chain. And these electrons are sort of like water going down a waterfall where they're moving to a lower gravitational position. And as these electrons are flowing, you can sort of picture then, let's say, NADH delivering the electrons to the ETC, the electrons flowing, and as those electrons flow, what happens is protons are pumped into the intermembrane space. Protons. Such that, picture this. I'm going to draw a really large H plus in the space between the inner and outer membrane, and a relatively small H plus in the matrix area. Are you with me? What do these differences in size represent?
That the inner membrane becomes more acidic and the other one becomes more basic.
That's right. This is becoming, with high acidity, more acidic, lower pH. This is becoming more basic with a higher pH. But what we're getting is that there's a concentration gradient of protons developed as we pump the protons into this space.
Jerry,
Yes?
the electron transport chain, okay, it's on the inner
Mitochondrial membrane.
Inner
Right,
mitochondrial membrane.
right.
That's right.
So how, I need a visual, is it like the whole thing, you know, around the,
I'm going to give you the
do
figure
you know
in
what
the
I
text.
mean?
I brought all these figures in, and then I got busted. Here it is right here.
So does it encircle the whole
24/8.
membrane?
So 24/8. Here's what I want to do. Okay.
Right, and I've seen that, but I mean, is it the
So
whole?
what I want to show you is, can you
thing?
see these purple blobs?
Mhm.
Those purple blobs are components of the electron transport chain. This is the phospholipid bilayer of the inner mitochondrial membrane. And what's happening as, here we have NADH, here we have FADH2, they're delivering their electrons. This yellow line represents the electron flow. Again, I apologize that we have, all our bulbs are out in the overhead projector. So I'm doing the best. But you have figure 24/8 in your text. And so as the electrons flow, the yellow line, eventually being grabbed by oxygen, by the way, the electrons, protons are pumped. Here, this big blue arrow, here, this big blue arrow, and here. So that electrons flow and protons are pumped into the inner membrane space. So we have several things happening that we can talk about. Again, I will have these images Monday, and I plan to start my lecture on Monday in Dogwood, pulling up those images in color, and I can walk that through as a part of review. Okay? But you folks should study those figures in your text. Studying those figures is a great way to learn this process.
But
Outstanding artwork.
are those pumps, so to speak?
Are those what?
Pumping, you know, that are pumping the protons out, et cetera, are they throughout the whole
Yes.
inner membrane
There's
that encircles the
thousands.
whole mitochondria?
There's thousands of these teams of electron transport chains in the inner mitochondrial
Okay,
membrane.
so
It's not just one, as I've shown ETC here. Can you picture it? ETC.
Mhm.
They're all over, aren't they?
Okay.
Does that help?
All right,
Each
that's
one of these
what I
then
wanted to
is
know.
a team of electron carriers. But again, in the figure, it only shows the one. So let's get back and talk about what's happening, because so the electrons are passed down. As the electrons are passed down, eventually those electrons are captured by oxygen. Oxygen is the final electron acceptor. It is sitting down at the bottom of the waterfall, gobbling up all the electrons. And what happens is the oxygen grabs the electrons, it grabs a couple of protons, and makes what? Water. Exactly. So O2 plus electrons plus protons makes H2O. Now actually we should put 1/2 of a molecule of oxygen gas plus a number of electrons plus 2 protons makes H2O. So that's what happens to the electrons. And oh, just checking. Yeah. Is it locked?
Yes.
Okay. So I'm just here till probably 3:15, teaching on Jerry Hodge. Where's the energy developing here? Because making water doesn't give us any energy, does it? Water is a freebie. We can get water elsewhere. Well, where the energy is developing is in this proton gradient. And in the figure in your text, it actually shows it like this. So I'm going to draw it the way the text kind of draws it. Another good way to show it. A bunch of pluses in here and a bunch of minuses here. Well, that's relative, isn't it? There's relatively more pluses